So our next speaker is Niamh MacSweeney. Niamh, you're here, I think. Yeah.
Yeah, I'm here. I'll just share my screen if that's okay.
And I think, did I see Heather Wally?
Yep, I'm here. Yeah.
Hi. Heather, hi. So will you do us an intro? Yes. Please.
Yes,
thanks.
Of course. So hello, everybody. Good morning. So it's a real pleasure to introduce Niamh this morning. So Niamh started with us at the University of Edinburgh in 2018. So she's in her final year this year. And she's coped admirably with all the chops and changes that have had to happen because of COVID. She's a real pleasure to supervise, and I'm not sure if she's going to speak today about her passion for open science as well as her interest in her research. So I'd just like to mention that Niamh was awarded a highly commended prize by the British Neuroscience Association Credibility Award for her work in open science and reproducibility. So well done to Niamh for that. And yes, I'll just pass over to her to give her presentation. Thanks, Niamh.
Great, thanks very much, Heather. So yeah, hi, my name is Niamh. I'm a final year PhD students. This is my last Scholars Day. So today I'm just going to give an overview of the work I've done to date in my PhD, and I suppose focus more on the research that I have undertaken in the past year. So it might be a bit of a whirlwind, but I'll try and stick to time and kind of talk you through what I've been up to. Okay, so the overarching aim of my PhD is to understand the biological and psychosocial factors associated with adolescent depression. And in the interest of time, I'm not going to go into too much background information on kind of adolescent depression. But just, I think there's kind of two main things that would be good to keep in mind. So first of all, adolescence is the time in our lives when we're most vulnerable to the onset of depression. And we know that depression is one of the leading causes of illness and disability in young people. And then secondly, during adolescence, the brain undergoes dramatic changes. So they reckon that adolescence is actually this second period of kind of heightened brain development, and the brain is changing as much as it is in kind of the newborn phase. So it really is a time of flux. And I suppose I'm approaching understanding adolescent depression with both of these things in mind. And that's very much shaped the approaches I've taken in my research. So my research is kind of broadly split into two main strands. So the first two are data analysis-based projects, and these emerged because of COVID. So actually prior to that, I was going to do an entirely data collection-based PhD, but that changed quite quickly when COVID happened. So I've been working on two studies with the ABCD data set, and then a third study looking at irritability in adolescents. So all of these adopt very much a neuroimaging lens and trying to understand the kind of root of depression in the developing brain and how that associates with biological and other psychosocial factors. So I'm going to kind of discuss each of these today. So just to give a bit of background so people know what kind of data I've been working with. So ABCD is a study based in the States. It stands for the Adolescent Brain and Cognitive Development Study. It's an absolutely fantastic resource, and it's available to all researchers. And it's a population-based study that is longitudinal. So they've recruited nearly 12,000 young people across the US when they were 9 and 10. And then they're going to follow these young people for the next 10 years. And they've tried to make the sample as demographically diverse as possible, which is super. And they've collected a host of variables. So they have neuroimaging data, and then an array of mental health, social and environmental data. So it really is a wonderful resource to the field, and will hopefully help us better understand the factors that shape development. So the first project in my PhD was looking at brain structural associations with depression in adolescents. And I'm going to just kind of briefly talk about this project because I went into quite a lot of detail at the last meeting. But just as an update, this paper was published in eClinical Medicine in November. And this is a project that I led with a postdoc in our group. And just to kind of summarize the main findings of the paper, so we were interested in what brain structures were associated with depression in adolescents, and whether or not this kind of related to the findings that we see in adult samples. Broadly speaking, we found that there were very similar findings to those seen in adult samples, so decreased cortical volume, as well as differences in white matter microstructure. So these are kind of the connections between different parts of the brain. So we see similarities between the two samples there, but also some notable differences, especially in terms of surface area. And these findings suggest that neuroanatomical differences may be present early in the disease course. So even though the young people that were kind of involved in kind of this, or the data that was available for this analysis, the young people were only aged 9 to 11, so at the very early stages of adolescence. So this study definitely like paves a lot of ways forward to study whether or not these brain structural associations change over adolescence or how they might develop. So definitely a lot of exciting avenues to follow up on there. So the second project that I've been working on in my PhD in terms of the ABCD work I've been doing relates to brain structure as well, but looking at pubertal timing and how that relates to an increased risk of depression. This project emerged out of the research that we did in project one. So just as kind of a brief background to kind of puberty and how it might relate to mental health risk. So we know that earlier pubertal timing is associated with higher rates of depression in adolescents. And beyond age-related changes, so as I mentioned earlier, we know that the brain undergoes dramatic change during adolescence. We also know that pubertal development specifically is associated with changes in brain areas related to cognitive control, emotion regulation, making. However, we're kind of where brain structure might be. So this is your pubertal development relative to your peers, and practice the aim of that study, which I've been working on for the past couple of months. So I'll just briefly kind of walk through the methods of the study. So this project is a registered report style article. So this is an open research kind of publishing format whereby your project undergoes peer review before you do your data analysis. I'm not going to go into too much more detail than that, just in the interest of time, but very happy to have a chat about doing a registered report as part of a PhD. I think it's been it's been a lot of work, but like a really good experience, and definitely like strengthened the study, because I'm currently working on revisions from reviewers on my data analysis plan. So I actually haven't done my main analysis yet, but it's been wonderful to get feedback at this stage. So we're using a smaller sample of ABCD just so that we have data from people for kind of our time points of interest. So we're looking to see, our first hypothesis is hypothesizing that earlier pubertal timing at year 1, so when the young people are aged 10 to 11, is associated with higher depressive symptoms at year 3, when the young people are aged 12 to 13. And then our second hypothesis is that the relationship between pubertal timing and depressive symptoms will be mediated by specific brain structures measured at year 2. So this is our mediation model setup, which will test using structural equation modeling, and then we'll use nested linear effects model to look at the other associations. And the regions of interest that we identified emerged from pilot we did use a sample of the ABCD study. Which I suppose is just kind of was very useful for our purposes, and I suppose just highlights one advantage of using large existing data sets, so that you can use a small sample of like 1,000 people at 10% of the sample to kind of identify and inform your hypotheses. So this is one that I'll continue working on at the moment, and hopefully submit next week submit the revisions next week. And then the third project I'd like to discuss briefly today is my data collection-based project. So this is quite different to the other research I've been discussing, in that rather than working with a sample of 12,000 people, we're working with a sample of 30. And these were young people based in Edinburgh that we recruited last year. So the kind of main aim of this study was to better understand the neural correlates of irritability in adolescent depression. So what I mean by this is we know that irritability is a core symptom of depression in adolescence, and it's also a risk factor for emotion regulation problems. But what's going on in the brain when young people are irritated isn't very well understood, and I think this is due to a lot of reasons. Irritability isn't just a symptom of depression; it co-occurs in lots of other mental health difficulties, and then there's a variety of measures that are used to try and study irritability. But kind of the main gap in the literature that we identified is that when people have tried to understand irritability in the past, the social context of irritability has been overlooked. So if you look at the neuroimaging studies, a lot of the time the participant is undergoes an MRI scan, and then they're asked to solve an impossible maths problem, and that's kind of the way of inducing an irritated state. And while that will probably make everyone frustrated, for our purposes, especially studying irritability within adolescence, the social context is so important, because a young person's experience of irritability is very much rooted in their social world. So what we wanted to do is to try and study irritability bearing this kind of social importance in mind, and we developed a kind of new fMRI task working with young people to develop this task so that it kind of reflected the lives of young people as much as possible. And then we piloted this in a small sample of young people. And so this analysis is kind of currently underway, but I'll kind of briefly walk you through what we found to date. So this is just an example of the kinds of scenarios that people will read when in the scanner. So I find it irritating when people tell me to do something even though I was going to do it. And this is just some kind of basic information on our sample. So this is just the analysis that we've done to date. So we adopted a graph theory approach. And essentially what this does is it models the brain as a network consisting of nodes and edges. So if you can see here in the diagram, you've got different nodes of kind of function, and then how they relate to other nodes within the brain. And then the edges are kind of the connections between these nodes. And you can use these network properties to kind of investigate different properties of these networks. So you can see how well they kind of talk to each other or how connected they are. And one approach that we took was to look at the kind of integration of these networks. And this is something that's measured using an approach or using a metric called clustering coefficient. So we found that higher irritability was associated with increased dominance of an area of the brain called the anterior cingulate cortex. So this is really important for emotion regulation. And we found that this had increased dominance over a fragmented network. So there was a kind of so if you see here in the diagram of the nodes and the edges, there were less triangles. So there was less kind of neighboring nodes. It was a fragmented network. So this kind of highlights the important role of this region and its connections in the kind of neurobiology of irritability. And this is very recent work that we've done over the last few weeks, and we're going to follow this up with some more analysis, looking at the kind of dynamic nature of brain activity in the context of irritability. So I know I'm kind of coming up to time, if not over time, but just to so yeah, as Heather mentioned, this is my last year of my PhD, so I'm due to submit at the end of the year. And my main priority over the next couple of months will just be finishing up the registered report and the irritability project and submitting them for publication. And also another kind of review paper that we've worked on with a young person, which is kind of, we're preparing for submission at the moment, as well as attending some conferences, and then submitting the thesis by the end of the year. And then just briefly mention some other activities which Mental Health UK helped support. So just to kind of highlight one, I suppose, which I was invited to visit Professor Christian Tamnis's group in Oslo last November, which was a fantastic opportunity, and was very thankful for MHR UK to support the trip. And then, as Heather mentioned, good news last week that from the BNA that I've been awarded highly commended in the student researcher category. So that was really nice, I suppose, as an opportunity to reflect on the open research that I've done in my PhD to date, and the how it's shaped my approach to research. So to finish, I'd like to say a huge to my supervisors and collaborators here in Scotland, and yeah, open up for any questions. Thanks very much.
Thanks, Niamh. Great stuff. Would you like to take down your yeah.
Sorry, I was a bit over time.
Don't worry, don't worry. We've got a bit of extra time because I didn't witter on at the beginning. So Peter, you've got your hand up.
Yes, thank you. I really enjoyed your talk, Niamh, but I just wondered whether you could tell us a little bit more about the registered report approach, because I think it's really important. And also the ABC study is open data, designed to be open. And before the registering of your report, how do you know, or how do you find out about whether other people around the globe are using the same data set to do something similar? And are you able to communicate with them and synthesize or compare and contrast your data?
Yeah, great question. Thanks very much, Peter. So to answer your first question, so yeah, I didn't go into too much detail about the registered report because I feel like you can give like a whole lecture on what a registered report is. But so for those of you that don't know, it's a publishing format that kind of promotes open science. So rather than having all of the peer review at the end of your article, it splits it into two parts. So what you do is you write your introduction and a very detailed methods and data analysis plan, and then that's submitted to a journal for peer review. So you get feedback on your proposed plans before you do your analysis. And then if you address their concerns, and from my experience, we got lots of really positive feedback on the paper and really helpful and constructive feedback, and kind of highlighted things that maybe we didn't think of, or kind of it's great because you get like other experts feedback on your project, and then they give you the opportunity to kind of shape it and strengthen the project. So I'm just kind of at that stage now and preparing to resubmit, and then hopefully I'll be issued something called an in-principle acceptance. So that means that regardless of what I find when I do my analysis, as long as I stick to my kind of registered plan, they'll publish my findings. So it's kind of a way that kind of, I suppose, gives the researcher reassurance that like even if your findings aren't significant, that they'll still be published. And I suppose it just kind of stops or kind of reduces the presence of p-hacking and hypothesizing after the results are known, and it's just a very transparent way of doing research. I think in terms of the experience of it, it's quite like a front-loaded workflow. So like you do a lot of work now, but I'm hoping that because you literally know exactly how you're going to do every aspect of your study, it will make the kind of data analysis part of the project a bit more straightforward. I will say that I think it's a lot easier to do a registered report with secondary data sets like ABCD, because having collected data as well, the plan you have at the start versus in terms of what data you're going to collect versus the sample you're actually able to recruit can be quite different, and that's through no fault of your own. I think that's just life as a researcher collecting data. Yeah, so I won't say much more on that, but the second point then in terms of ABCD. So yeah, I think when you work with a freely available data set like ABCD, there's always going to be risk of overlap. I think ABCD have done a very good job at documenting who's researching what. So they really encourage all researchers to pre-register their analysis and try and do registered reports if you can. On their website, they have a list of all of the studies that have already been published, and then they also encourage people to upload kind of protocols to the open science framework and things like that. And I didn't go into detail in this in my presentation, but I was part of a kind of ABCD summer school last year that was kind of highlighting collaboration and kind of working together. So they really have tried to make sure that everyone's working together rather than being super competitive. And we've actually written a protocol paper together on that, which kind of is a user guide for ABCD. So that was linked to my slides. So maybe they'll be available afterwards.
Sounds a really important experience in your PhD studies. Thanks.
Yeah, thanks. Sorry, that was a very long answer.
No, that's okay. Alarmingly, I see that this session is ending in 20 seconds. I it's a bit earlier than I anticipated, but I think there might have been some behind the scenes maneuvering, which I only, but thank you very much, Niamh, for that talk. It was really excellent to see your work, and thank you to all the speakers.